Hi everyone, thank you for coming today. Shahid, yes, if you can say something so we can test the audio. Hi, can you please say something? I'm getting a lot of noise, but I'm not getting any speech from you. Shahid, we're getting a lot of noise, but nothing else. Can you please change your microphone? I can see that you unmuted, but there is nothing. I can hear nothing except a bit of noise. Do you mind changing your microphone, please? Apologies, everyone. We're just going to try and sort out the technical issues we're having, and we'll be with you very shortly. Hi, everyone. Sorry about the delay. If you can be patient for a few more moments, please. Okay, let's try this again. Shahid, do you mind unplugging yourself and
Yeah.
yes, there we go.
Okay. Okay, perfect. Thank you. Sorry about the delay. Okay. So hello, everyone, and thank you to the research app team for inviting me to present this important topic on nephrotoxic nephritis, glomerulonephritis, animal model versus human disease, and this is a presentation in collaboration and linking with the Renal Patient Support Group, the RPSG, and in support of World Kidney Day 2022. And the main theme of World Kidney Day this year, just to highlight, is around kidney care for all, and that's slide one. I'm now moving on to slide 2. So a bit of introduction about this topic. So research with animal models has shown the crucial interaction between certain inflammatory cells and cells intrinsic to the kidney that is both fundamental and unique to the pathogenesis or the pathology of glomerulonephritis. Now, the term glomerulonephritis basically encompasses a range of immune-mediated disorders that cause inflammation, and the main terminology is where glomerulonephritis is concerned, inflammation is linked with that, and within the glomerulus, which are the functional components of the kidneys, and the mechanism of interaction between these cells and the mediators of their coordinated response to inflammation are being elucidated. So despite these pathophysiological advances and in areas of laboratory practice and pathology, treatments for glomerulonephritis remain non-specific, can be hazardous, and only partly successful. Now, therefore, glomerulonephritis and glomerulonephritis throughout this presentation will be abbreviated as GN. Therefore, GN remains a common cause of chronic kidney disease globally. So moving on to slide 3. So here, I'm going to just talk a little bit about the mechanisms. Now, the mechanisms in this context is where evidence of complement is something in relating to the immune system, and in relating to parts of the immune system where it's linked with immune related diseases. So evidence of complement deposition within the glomerulus is a key feature, or is one of the main components that forms a glomerulonephritis, or GN, and the role of complement in this context is implied by several observations in human disease. Now, indirect evidence obtained from human studies has been supported by findings in animal models. And now, when we're referring to animal models, and there will be a slide around this, but just a heads up, when in related to animal models overall, in relating to kidney care and treatments, and relating to different kinds of treatments, the main animal models are murine type mice models, or rat type animal models. But we'll get into that in the next few slides. So the mechanisms by which macrophages mediate renal injury remains to be confirmed, but it is likely to include promotion of inflammation via release of pro-inflammatory cytokines such as tumor necrosis factor, or TNF, and recruitment of macrophages and other leukocytes or white cells through or via release of chemotactic molecules. Now, chemotactic molecules include a monocyte chemotactic protein, protein type 1 or MCP type 1 and cell proliferation by release of growth factors such as macrophage colony stimulating factor, which is an abbreviated as MCSF. And these have been established. Now, cell death as part of the mechanisms and in relating to GN, cell death occurs through the release of reactive oxygen species, including nitric oxide, and control of fibrosis and repair via release of various enzymes. And now I'm going to move on to the next slide, slide 4. So a little bit of epidemiology and background relating to a GN, and in this context relating to in bringing understanding around the animal models and human disease aspect a bit more. Now, from an epidemiological or statistical basis, population-based screening studies have shown that evidence of kidney damage by protein urea, which is referred to as where there's protein leaking through the kidneys, hematuria, which is basically where there is evidence of blood which is being voided through urine, which is not usual, although calculated glomerular filtration rate or GFR, or a combination of these features is present in about 16% or just less than 20% of adults in Australia, and a similar proportion in the USA. Now, this is there will be a list of references and sources at the end of this particular presentation. So I will highlight where more understanding can be sought through the literature around this particular context. Accurate classification requires a histological diagnosis, and consequently the reported prevalence might vary according to local indications for renal biopsy. Now, renal biopsy, as some may know, for example, is a basis to try and identify different forms of scarring, or why, for example, someone may have a form of glomerulonephritis, and why glomerulonephritis has occurred. Now, a retrospective analysis of all renal biopsies performed between 1995 and 1997 in the Australian state of Victoria during 1995 and 1997 found that around 21 and a half or 22 people, around about 22 people per 100,000 population per year underwent a form of renal biopsy, yielding an annual incidence of biopsy proven GN of 12.3 per 100,000 in the population. So on a sort of from an epidemiological or population health perspective, basically almost 10%, or sorry, just over 10% of the population, which might not seem a lot, had a form or were at risk of having a form of glomerulonephritis. Now, the most commonly diagnosed types of GN in adults were Iga Nephropathy, IgA nephropathy, something referred to also as focal and segmental glomerulosclerosis. Now, and also vasculitis. Those most commonly diagnosed are actually in young people, and pediatrics and young people were minimal. were also forms relating to minimal change disease or minimal change focal and segmental glomerulosclerosis, lupus nephritis, and also IgA nephropathy. Now, when you add up the different forms of GN, then GN across population, basically you can see that generally speaking, there is a kind of misnomer when looking at GN across different populations, and so the statistics are pretty preliminary, and what clinicians and scientists don't really know is the full extent of GN, globally. But we know that it is a problem. So now moving on to slide 5. Now, a little bit about the pathogenesis. So the pathogenesis of GN is quite complex. The intricacy arises from factors such as autoimmunity, so where particular immune cells, as part of physiology, attack itself, attack or cause a prompt to cause an immune response, so that the cells cause are doing more harm than being protective. So factors such as autoimmunity, cancer, and structural abnormalities within the kidney or kidneys. Now, there is variability in the susceptibility to GN, which is likely to have a genetic basis as well. And there are also complex interactions between soluble factors, including antibodies, complement, chemokines, cytokines, and growth factors. Now, all of these certain terminologies I mentioned, for example, antibodies, complement, chemokines, and cytokines, and growth factors, generally in the realm of research relating to kidney care practice or nephrology and scientific or laboratory practices. These particular cells are perhaps the most prominent cell types that are studied under close investigation when looking at GN, and also where I mentioned, for example, different forms of other cell types. For example, those two, for example, MCF. Or MC, and also MCP type 1 and other cell types, all are closely linked not just to GN but other forms of kidney disease. So generally speaking, when investigating, when researchers and scientists and clinicians are investigating around different forms of kidney disease and not just GN, generally the pathogenesis relate very much to these cells being under close investigation all the time. Now, in line with this, the contribution of non-specific factors such as hypertension or high blood pressure or proteinuria or protein leaking promote continuing kidney damage. Now, protein leaking is a very primitive a link to where kidney disease can occur. Now, the issue with proteinuria is not so much in the first place where there's protein leaking through the urea. That's not much. That in the first instance, that's not the main issue. The main issue is actually when there is protein leaking or proteinuria, there normally is or can be reflux that is attached to that, and that when reflux happens, it's then where patients, especially those with, for example, weaker immune systems, where they have reflux, and that can cause damage to the urinary tract, and that is why proteinuria can be problematic. Now, generally, despite despite the sort of some of the advances in certain aspects around pharmacotherapy, for example, there can be continuing kidney damage or renal damage despite resolution of the initial insult. And a constant feature of GN is the presence of as I briefly mentioned, inflammatory leukocytes, inflammatory cells, including leukocytes within the glomerulus of the kidneys and the interstitial compartments of the kidney. Now, macrophages are important again, and T cells or T lymphocytes are also important in relating to the dominance they play in the pathogenesis. Now, they're important because these cell types are not just present, but they also have there is also intensity of how their presence cause cellular damage in relating to how these cell types can accumulate where GN exists, particularly with the interstitium, rather than the glomerulus. So where GN is concerned, it's primarily the glomerulus is the main part, or the main area that is affected. But with the glomerulus, there are other areas that extend part of that as well. So basically, GN is where there is the first basis of where this disease can occur. Where insult and injury can occur, but then thereafter there is problems where other areas of other functional parts of the kidneys are also are susceptible to damage. And so along with the interstitium, rather than the glomerulus, has repeatedly been found in research to correlate with the clinical and pathological severity of disease in animal models and in human beings. So there's a link between those two focuses. Now, I'm going to move on to slide 6. So a little bit about the actual basis around the animal models now. Now, 3 main points here on this slide. Number one, animal models are used during research and investigation of human diseases. The animals selected will usually meet a determined taxonomic equivalency to humans in order to react to disease or its treatment in a way that resembles human physiology. So, for example, let's say, if someone is studying or looking at disease of let's say the liver, let's say we're doing looking at the liver, and then looking at the liver in animals and in humans. Basically, there's no point in looking at a bird, for example, a liver's bird, and trying to compare and link that to human beings when looking at liver disease in humans. So the same is the same approach when looking at kidneys, and in this case looking at glomerulonephritis. So basically, in relating to this, point 2, research relating to nephrotoxic nephritis and glomerulonephritis can be traced back to the 1960s and actually the late 1970s, when observations were reported on the role of complement in NTN produced in the rat also by the rat and rabbits or duck nephrotoxic sera. So what this means is that when when looking at looking at animal models and human disease, GN is the actual human disease. So it's the glomerulonephritis. It's the human disease. Nephrotoxic nephritis is what brings on is what is part of the onset of what triggers GN. So there's lots of triggers that relate that tie into or part of NTN or nephrotoxic nephritis. So I just want listeners to get a feel for that understanding, because basically in different animal models, so I've mentioned, for example, in rabbit also, and in duck, for example, the sera in the sera or the the blood that are within these particular animals are susceptible to nephrotoxicity happening, and thus because of how the pathway of how nephrotoxic nephritis occurs or potentially is can take place within those particular animals as well. Basically, when studying back in the 1960s and the 1970s, researchers and investigators actually were able to use more than the kinds of models, animal models that exist that are out there at the moment. So that in some contexts that there was a bit more, let's say, there was a bit more there was a bit more of a looser approach in relating to studying different diseases and linking and how animal models would link to those diseases back in the 1960s and early and mid 70s. And so point 3 in this, in addition, the course of complement or the immune cells and the reactions in the glomerulus in NTN is in nephrotoxic nephritis is used as a measure of the course of immunological events that relate and occur as part of GN or glomerulonephritis. So I'm going to move on to slide 7 now. So a little bit more into the animal models, and I want to get this understanding across, because with animal models, basically, animal models are used in lots of different clinical trials as can be used in clinical trials in relating to how humans with particular diseases can benefit from treatments, and before implementing any treatments, obviously in human beings, tests and investigations can be done in certain animal models, and clinical trials are part of this. Animal models, a bit more here then. Animal models of GN closely replicate features of human GN, as mentioned, and have provided insights into the disease. The reproducibility and uniformity of such animal models has also permitted a step-by-step dissection of many of the mechanisms involved. Now, nephrotoxic nephritis, or NTN, is produced experimentally by the intravenous injection of antibody directed against a more specific area of the glomerulus, something known as the glomerular basement membrane, or GMB, antigens of the recipient species. So basically, from the particular animal models I've mentioned, some of the animal models I've mentioned, some of the sera of those animal models can be injected into other humans in a way to understand whether treatment is going to work or not. And I can work the other way, by the way, and it's basically because if, for example, treatments aren't working in humans, then basically the recipient of the antibody that is should be used to treat can be used to understand, at least from clinical trial perspectives, that should not be used in humans to treat treat a GN in this case. So most of the accumulated inflammatory cells in many forms of human and experimental GN are macrophages. So hematology plays a significant role in relating to in the inflammatory processes of what takes place when there is when GN occurs. So slide 8 now. So a little bit about animal models, and the main one I'm going to talk about on this slide is the murine model, or the mice models. Now there's lots when certain tests and investigations are done according to researchers in laboratory practices in laboratory science and laboratory medicine or laboratory practice. Basically, there's lots of different mice or mouse models that can be used. Now, in relating to nephrotoxic nephritis and GN, there's one particular mouse or mice murine model that had actually worked really well, and this is a murine or mouse model known as the BALB C murine models. And basically these particular murine models have clarified the pathogenesis of GN of GN and several other renal related diseases when looking at GN, but also understanding the toxicology around specific markers of disease in relating to kidney damage. So if it's not GN, for example, the same murine models or mouse models can be used or implemented to understand how other diseases can occur. So the murine mouse models actually and the BALB C murine model in particular is very close to the human kidneys in some contexts under controlled conditions in a laboratory manner. Now, in retrospective research, 6-week-old female BALB C murine models were induced with injections of 50 milligrams or around 0.2 mils of bacteria lipopolysaccharides from Salmonella Minnesota twice a week for 5 weeks by the intraperitoneal route. So basically, in these mice models or murine models, what was happening, injections of bacterial lipopolysaccharides, which relate to understanding really how certain tissue that link to link to GN as a disease as well, how bacterial lipopolysaccharides from Salmonella were induced through the peritoneal or intraperitoneal route of those mice models. So the intraperitoneal area is very close to where near where the kidneys are. Basically, they were injected with bacterial lipopolysaccharides near the kidneys, or so that the investigators could understand what was happening around around the peritoneum. Now, in all murine models, given injections, features of something known as polyclonal B cell activation, or PBA, and GN developed. Now, this is important because it shows that what the investigators were doing to understand how nephrotoxic nephritis might occur, and how GN, and again how polyclonal B cell activation occurs. By that happening, basically it shows that when it comes to treatment, that if they know that that nephrotoxic nephritis is being induced successfully and GN does occur, then it also allows them as investigators to understand how treatment can be implemented in the same way, by understanding those immunological pathways. Now, again, as I mentioned, some of those cell types that I mentioned in one of the previous slides, some of those slides, for example, chemokines or cytokines, those would have acted up, which would have stirred on, for example, leukocytes or T lymphocytes to actually prompt a response or elicit a response. Now, the animal model, at least from murine mouse models, presents features that are often encountered in human GN, particularly those associated with bacterial infection. That's very important. If anyone takes anything from this slide set today, then know that is basically to say that the animal models that present features that are encountered that take place in the physiology of animal models, again like murine models, are encountered in human GN, and basically where there is a bacterial infection, the pathway around how bacterial infection occurs, or infection occurs in murine models, is actually not that far off as it takes place in relating to humans and human disease. Now, as well as autoimmune diseases such as something referred to known as systemic lupus erythematosus, or SLE. Now, the next slide, slide 9, I'm going to talk a little bit about rat models. Now, rat models work a little bit differently, and I'm gonna delve into a little bit why they're a little bit different. Now, there is a rat model known as the WK rat strain, which is also highly susceptible to experimental models of something relating to GN, very close to GN, and something which is known as crescentic glomerulonephritis. So CRGN. So it is GN, but where crescentic, where I mentioned the terminology crescentic, is basically a more specific area around the glomerulus again, where inflammation occurs. So it's not just general glomerulonephritis or general inflammation around the glomeruli, but more specific areas where there is a kind of a shaping which relates to the relating to the anatomy and of the kidneys, and this is something in anatomy and in textbooks known as crescentic glomerulonephritis, and this also relates to including nephrotoxic nephritis and experimental autoimmune glomerulonephritis, which is abbreviated as EAG. Now, these rats, or the WKY rats, or the rat strains, show development of NTN phenotypes. So there's different types of NTN that can occur in WKY rat, in rat strains, and that's not unusual. So the rats show development of NTN phenotypes, including including glomerular crescents around the crescentic aspects of the glomeruli and the kidneys, but more recently, CRGN, or crescentic glomerulonephritis, has been induced on a normally resistant rat genetic background, and identifies something known as a Lewis strain on two genetic areas, in two different genetic areas, CRGN1 and CRGN2, as new and potentially valuable model for NTN GN. So what this means, that there are two areas relating to the genetic around the rats, and the genetic basis and background, known again as the Lewis strain, as to how researchers and investigators can replicate NTN in rats, as well as in mice murine models. Now, this is important because it actually allows a little bit more diversity to how treatment can be implemented in understanding how to rid of issues around glomerulonephritis, at least to some extent. Not the full extent, because we must understand, when glomerulonephritis occurs, or when it happens, generally what happens is that other parts of the renal or kidney anatomy can also be affected, which means that GN is not always restricted to just the glomerulus. Now, the another point I want to make here is that the nephrotoxic serum nephritis, something that's related to NTN, so nephrotoxic serum nephritis, WKY rat, has also been used in research around that as well. Macrophages and T lymphocytes are the dominant cell types present, and the intensity of cellular accumulation, particular within the institution rather than the glomerulus itself, has been found to correlate with the clinical and pathological severity of disease in animal models and in humans. So I'm not going to move on to slide 10. So a little bit about the role of chemokines, and it's very important to get a kind of a basis around chemokines and cytokines, because again, where nephrotoxic nephritis is concerned, and where glomerulonephritis, or GN, is concerned, generally speaking, researchers, investigators, where laboratory practice is concerned, will focus primarily and quite intensely around chemokines or cytokines, because these immune cells can act as particular biomarkers of disease as well. So investigators looking into the immunology around this particular disease can use particular laboratory equipment or apparatus to understand, for example, where there is certain cell migration, or T cell, for example, those cells that prompt an attack in relating to immunological responses, actually can understand how T cells or white cells, for example, can actually prompt prompt an attack on foreign body to actually cause more devastation to organs, and certain laboratory equipment or apparatus can be used to understand T cell migration patterns, and where there is more risk or susceptibility of problems around where treatments might work or might not work, for example. So the role of chemokines are thus: the migration of inflammatory cells into an extravascular site requires a series of coordinated signals, including the generation of chemotactic gradient by the cells of the extravascular compartment. Now, the nature of the stimulus and the subsequent the subsequent spectrum of chemotactic factors produced determine the specific leukocyte or lymphocyte population recruited to the inflammatory cell. So this is what I mean when there is T cell migration. Now the main immune cells that require that everyone should get an understanding around is, for example, the CD8, for example, CD8 T cytotoxic CD8 cytotoxic T cells. Now these, when an inflammatory response occurs, this can cause problems where basically lots of other hematological cell types as well, for example, or parameters, actually then cause more devastation to anything that is foreign. And it's the migration patterns that are the migration patterns that are very important. So members of the chemokine family play a central role in this process by attracting and stimulating specific subsets of lymphocytes. Now, again, specific subsets can would include the CD8 T cells and CD4 T cells. Now, CD4 T cells are known to be related to T helper cells, but T helper cells or CD4 cells are basically a kind of a preliminary T cell type, where basically they lay ready basically to prompt CD8 T cells to actually launch an attack. It's a bit like saying, for example, in a very broad brush way, say for example, a person doesn't feel very comfortable with another person in front of them, but instead of approaching them directly, they would go to another mutual friend that they both know and say, can basically you tell that person to kind of leave me alone. So it's kind of a similar example in that context. Now animals genetically deficient in chemokines are resistant to disease induction, although results have not been uniform. They're not representable in some contexts, and with an unexpected acquisition of experimental anti-glomerular basement membrane disease seen or identifying in murine models deficient of what is known as chemokine receptors, or CCR type 1, a receptor for something that is known as RANTES and macrophage inflammatory protein type 1, and chemokines are thus identified as important in murine models and clearly active in human disease. So this is very important, because apart from the nephrotoxic nephritis or nephrotoxic NTN sera that can be identified in different models, chemokines in this context are additionally important because they are used to help identify how they are used or are used as a basis or a marker where there is perhaps more associate more from an anatomical perspective or an immune perspective. To some extent, there is more of a link between mice or murine models when looking at disease in human beings. So the next slide, slide 10, slide 11, and we haven't got many more. A bit more specifics around the role of chemokines in this slide. So intriguingly, one team had generated CXCR3, which is a chemokine receptor deficient murine models to investigate the functional role of CXCR3 in kidneys under pathological conditions. Now, CXCR3 is perhaps when CXCR3 as a chemokine receptor is actually one of the most studied receptors relating to an immune response, when especially where, for example, people who have had transplants, and I thought this would be a good point to talk a little bit about the premise of World Kidney Day in some context as well, and that is where CXCR3 is one of the main receptors that are actually investigated when someone has a transplant. For example, CXCR3 is actually investigated to understand where individuals who've had a transplant, where for example, their immunosuppression or the certain anti-rejection medicines that they may be prescribed, where if they're not working very well, or not or they're not they have not been dosed accordingly to the appropriate trial and toxicity levels, then what happens is CXCR3 can actually be traced through laboratory in laboratory practice, where basically they can, if there's sort of a movement to cause an attack on a transplanted organs, CXCR3 is part of what can be established as a marker, not just as a marker of disease, but also can be identified as one of the key biomarkers that are premised to have launched the anti-rejection process. Now, in nephrotoxic nephritis, this has been induced to analyze the molecular mechanisms of T cell recruitment in GN by CXCR3 and its 3 ligands. Now, there's something called IP10, there's CXCL10, and there's MIG and CXCL9, and there's ITAC, and there's CXCL11. And to identify potential new targets for therapeutic interventions. Now, these particular sort of mechanisms relating to therapeutic interventions, these are more specific, not so much in for example, anti-rejection treatment, relating to those individuals after these particular immune markers and ligands are actually more specific in preventing in some way or another GN, and So research has identified that in line with this, chemokine receptor CXCR3 mediates renal T cell recruitment and subsequent tissue injury in NTN. So CXCR3 is actually quite an important receptor when looking at tissue injury, looking at nephrotoxic nephritis, and looking at GN, looking at GN, and basically where investigators can understand what if so. For example, if the tests and investigations are reproducible or can be done across a different time lengths of a particular investigation, looking at human disease, a G or GN in this context, and looking at GN and nephrotoxic nephritis. For example, if the studies are reproducible, then basically it can investigators can actually implement they can understand where tissue injury occurs owing to specifically to NTN, but also know what sort of treatments can prevent that. And some of the treatments out there can prevent, in some contexts, can also prevent, for example, some forms of IgA. In some contexts, IgA nephropathy, proteinuria, and also prevents more detrimental effects of glomerulosclerosis. So the study provided a rationale for considering CXCR3 blockade in type 1 mediated renal inflammation as a future therapeutic option for patients with GN and GN subsets. Now, I'm just going to move on to slide 12. A few points here, and these are the main conclusions now. So we're pretty much there. So point 1 here is the review. This review or this work aimed to provide insight into a suitable model of NTN, nephrotoxic nephritis or GN, and relate its experimental validity in humans. Now, in relating to this, animal models with experimental NTN, GN are homogeneous, whereas patients and human GN are heterogeneous. So what this means is that when studying NTN GN under laboratory conditions, because what is being investigated under laboratory conditions is kind of a controlled sort of atmosphere, versus GN actually taking place in humans, causing glomerulonephritis and damage. Basically, in this regard, in humans, GN is always different. There's always potentially a different kind of causation as to why GN has occurred. Now, experimental disease is initiated at a time determined by researchers and investigators and runs a predictable course. So again, as I mentioned, under laboratory conditions, it's a controlled environment, so a controlled atmosphere. Now, thereby permitting intervention at different stages, including before induction, during disease, and during disease establishment, or late in the disease course. So patients typically have established established disease before treatment can be actually administered or given. And point two here is renal damage or kidney damage tends to be progressive. And make no mistake here, listeners, and that is to say that, for example, with renal damage and GN in particular, scarring around the kidneys, for example, may in some individuals may be incremental. In some stages, it may be basically a full attack to cause scarring around the kidneys, so that basically individuals may not be able to void urine to rid of toxins in the blood. So basically, renal damage can be progressive. It might not be. It may be incremental as well. So in the longer term, despite elimination of original source of injury. So even when the preliminary issue that is causing the injury or the GN, and apart from NTN, for example, where toxicity is related, basically, in relating to this, it could well be that the original source of the injury may be removed. It might be removed. It might be identified, but at the same time, the injury can be extensive to the fact that because of the sensitivity of what the kidneys are like, because they are vulnerable organs by the very nature of their pathology, then generally speaking, it can cause problems to individuals in the longer term. Now, differences between experimental and human GN complicate translation of experimental therapies into practice, and I'm sure listeners are aware of that. But stem cell based therapies can or should be explored more intricately, and so these these could include genetic modification to produce protective molecules within the kidneys, or to promote regeneration of damaged intrinsic kidney cells. Now, a side note on this is that the kidneys, as generally speaking, the cells of the kidneys, the cells and the tissue around the kidneys do not have they cannot regenerate naturally. So in clinical trials, there have been experiments and investigations to see what can be done to see if there is a way that certain cells within the kidneys can regenerate, so thus allowing longevity to the functional aspects of the kidneys overall. Now, the third and final point I want to make here, as part of this conclusion, is there is also a need for more up-to-date research on this topic. Now, NTN and nephrotoxic nephritis is quite an old subject in some contexts. So if anyone was to look at nephrotoxic nephritis now, the studies around NTN are not very detailed as much as they once were. So looking at this at NTN may be difficult. Any more recent literature around NTN nephrotoxic nephritis may be difficult to identify. However, there in this regard around this is that there is a requirement for further work further work is required to translate animal model research in order to achieve a better or a smarter understanding of GN disease in humans, and the complexity of GN research makes findings a challenge to replicate consistently. Now, again, in certain lab conditions, laboratory conditions, for example, and laboratory conditions, and again, murine in murine or mouse models versus the WKY rat models. Generally speaking, these kind of models generally have been used to actually study other forms of kidney disease, more so in some regard. So generally, again, understanding GN disease in humans is more there's more scope of looking at disease and treatment when linked to other forms of kidney disease, and not specifically always GN. So that can be a confounding factor, and it can cause issues. Now, the complexity of GN research also makes findings a challenge to replicate not just consistently, but consistently be one of the main things, but also making can be challenging for investigators to actually bring about bringing about sort of novelty around specific areas of kidney research, so that new treatments can be implemented. Now, finally, around this, there must be, or there should be, a continuing evidence-based assessment of immune and non-immune therapies in order to provide sort of the rational care for patients who have or who are susceptible or at risk of having GN. So that is the overall understanding around this particular topic. I am just gonna read a few of the references in the next slide 13, and this is basically where I'm just going to highlight a few main references is here. So you have there's work by Chadban and team from Chadban and Atkins, who have a paper who published around this topic, glomerulonephritis, and it's a little bit dated. It's work that was dated in the Lancet journal from 2005. There is also works by Brown and team in the late 90s, in the late noughties, in 96, where they looked at the regulation and expression of chemokines' potential role in glomerulonephritis. There's work by Wader and team, the role of chemokines in glomerulonephritis, and that was published in the Frontiers in Biosciences in 2008, I believe. There's quite a few. There's works by a kind of a renowned author around nephrotoxic nephritis, and that's someone known as Anu, and he's this is someone who's published on experimental glomerulonephritis, and looking at the participation of complement in nephrotoxic nephritis, and this is work that has been published in the journal Experimental Medicine, and it's actually really quite a dated piece of work, but actually one of the most primary the primary authors, and one of the main kind of founding sort of authors that looked at nephrotoxic nephritis in the early 60s. So this is a publication that's dating back since 1960 in 1964. Again, published in the Journal of Experimental Medicine, and you published quite a few around this particular topic in the same journal, and I won't read all of them. And then there's the final one I am going to read is a paper by D'Souza and that person, that author's colleagues who have published a work in 2013 looking at experimental crescentic glomerulonephritis, a new bio-oncogenic rat model, and this was published in I believe in the Journal of Disease and Model Mechanisms. And so that closes this presentation. And just one final slide. I have done this presentation on behalf of the Renal Patient Support Group, or the RPSG, and linking with World Kidney Day 2022, where the theme again was around kidney care for all. So on the final slide, I do want to highlight some acknowledgements and relating to this work, and this work is dedicated to dear and departed friends, and I'm going to highlight just a few of these. And these are people who have passed away with different forms of kidney disease, or chronic kidney disease, and this work is to be linked and acknowledges them. So there's Ian Purchase, Emma Markwick, Cephal Kirbish, Ratna Das, Maureen Omondi, Everton Barkley, Carl Luton, Margaret Cromley, Christine Gaynor, David Stevens, Philip Starr, Rob Burney, Rob Argue, Leanna Bruce Kidman, and many others who have lost their lives to chronic kidney disease over the years. So I just want to say thank you once again to the researcher app team for inviting this collaborative effort on behalf of the Renal Patient Support Group and in support of World Kidney Day 2022. Thank you for taking the time to listen.
Thank you, Shahid. That was really interesting, and we're looking forward to your presentation tomorrow. So just to remind everyone, we have a second part of this presentation tomorrow at 4 p.m. GMT. So I hope to see you all there.
Thank you very much. Thank you. Okay.
Thank you. Bye-bye.
Okay, bye now. Bye.